So it's my pleasure to announce our next speaker, Dr. Patricia Dickson. Again, third in a row of physicians that has treated my family and that we think a lot of as well. Dr. Dickson is the Centennial Professor of Pediatrics and Chief of the Division of Genetics and Genomic Medicine at Washington University School of Medicine in St. Louis. She previously worked at Harbor UCLA Medical Center in Los Angeles County. She is a member of the steering committee for the Hope Center for Neurological Disorders and a co-investigator in Washington University's Intellectual and Development Disabilities Research Center. She also serves on the Society Scientific Advisory Board, and she is going to speak on gene therapy for mucolipidosis. You know, we talk about the MPSs a lot at the National MPS Society, but we also cover the mucolipidosis as well. And it's a little bit different kind of a disorder in that many different enzymes are impaired and impacted, which makes the theory has always been the treatment harder for that. And we're so thrilled that we're finally to a point where there's some good science going on and potential treatment for that. So welcome, Dr. Dickson.
Thank you so much, Steve. I'm checking to see if the pointer works over here because I understand that the people in the room on the other side won't be able to see the pointer with the clicker, but I don't think the laser pointer transfers over to the screen. No, okay. I will do my best to describe what I am pointing at. So I am talking about mucolipidosis types 2 and 3. So mucolipidosis types 2 and 3 are caused by deficiency of an enzyme. The name is glycnic-1 phosphotransferase, but we refer to it as PTAs for short. And this is not a lysosomal enzyme. So you can't replace it like you can for some of the other MPS disorders where you can give an enzyme replacement therapy. This enzyme lives in the Golgi complex, the Golgi apparatus, where it puts mannose-6-phosphate onto the lysosomal enzyme so that they traffic normally to the lysosome and can help the disease there. The gene responsible is GNPTAB. And in mucolipidosis types 2 and 3, you have a deficiency of mannose-6-phosphate on essentially all of your lysosomal enzymes. There's a severe form that's known as I cell disease, or ML2. Attenuated forms are ML3, alpha beta, or gamma. There's a gamma subunit of this PTAs that's encoded by a different gene. So S1S3 is a truncated form of the natural PTAs. So it has sequences from the natural PTAs, but S1S3 is not something that your body normally makes. It is, however, a shorter gene. It fits more easily into viral packaging, such as adeno-associated virus, and it is more efficient than the natural PTAs. So we have been working with M6P Therapeutics and the Cornfield Lab. They have helped us make a gene therapy using an adeno-associated virus. It has S1S3 in it and a promoter, and we have been administering these AAV9 vectors to mucolipidosis type 2 mice that are knockout null for the PTAs, GNPTAB, the alpha beta subunits of the PTAs. And giving them intravenous dosing of the gene therapy at 4 weeks of age and studying them throughout the course of their life. So I'll present those data here. And if you were at one of the previous meetings, this part of the talk may be familiar, but first, we looked at whether you could restore the mannose-6-phosphate content of lysosomal enzymes using this approach. So this graph shows the binding to the mannose-6-phosphate receptor, which is our assay. You are not seeing the slides advance. You are not seeing this because I am pressing it one place and not on the other place. Okay. Right. So let me go back on my computer. Okay, and forward on the, okay. So This graph shows the manna-6-phosphate content of different lysosomal enzymes in the serum of the treated mice. So A is beta-hexosaminidase, B is alpha-mannacidase, C is beta-mannacidase. And they're at different enzyme concentrations. So you can see that in when there's not treatment, or in the ML2 mice, that they have very low levels of manna-6-phosphate. And then this is mice serum at 2 weeks, 6 weeks, 10 weeks, and 14 weeks after treatment. This is another way of looking at the data. You can see that wild type levels of enzyme generally in the serum are pretty low. One of the features of ML2 and ML3 disease is that you see elevated levels of lysosomal enzymes in serum. In the ML2 mice, however, the amount of those enzymes that have manna-6-phosphate on them is very, very low. In the treated mice, you see elevated levels of these enzymes, but these are the levels of manna-6-phosphorylated lysosomal enzymes. So you have elevated levels of circulating lysosomal enzymes in serum that have manna-6-phosphate in the treated mice. This is a depiction of the mRNA, or the transcription of the S1S3 phosphotransferase, or the expression of it. On the left here is the mRNA copies in liver, heart, and brain. Those are these middle bars in the treated mice versus the carrier vehicle treated and the untreated ML2 mice. And the pancreas and salivary gland. All of the y-axes are different here. And of note, the brain, if you look very closely, the y-axes, these are very, very low levels. In fact, the brain level was basically the same as the untreated mice. But in the other tissues, we saw very good levels. And here is what we call our in situ hybridization, or the expression by RNA hybridization in tissues in the liver and the brown adipose tissue near the salivary glands. This is the red markings here showing the S1S3 at the mice even 50 weeks, so a year after the dose, you can still see expression. The histology improves partially with this treatment, and this is at 50 weeks. So again, this is a year out, so a very long time post-treatment. We're looking at the submandibular salivary gland on the top row, parotid salivary gland in the middle row, and then on the pancreas. The center column are the untreated ML2 mice, and you can see the vacuoles. These are the tissues that in the mice have a lot of storage. In the treated mice, you see a slight reduction. It's most obvious here in the pancreas of the lysosomal storage of the treated mice. Behavior improved partially in the mice. Notably, we found that the mice have reduced visual acuity. They have a retinopathy that seems to be unique to the mouse model. We did see that this retinopathy, or at least that the vision improved. This is in the S1S3 treated mice compared to the ML2 control. Let's see. Okay. We did not see substantial differences between the treated mice and the untreated ML2 mice in terms of weight, length, bone mineral density, and bone mineral content. And we did see an increase in survival. It was about a 12-week or a 3-month increase in survival in the treated ML2 mice compared to the knockout ML2 mice. So at the end of that experiment, we concluded that the AAV-S1S3 administered intravenously to the adult mice resulted in long-term expression, restored the manna-6 phosphorylation content of some serum lysosomal enzymes, and partially improved pathology. We saw incomplete correction of the phenotype, and we thought that potentially we might improve this with earlier initiation of therapy or an intracerebroventricular administration, so that we could get more of the PTase enzyme into the brain. So the first thing we did was a pilot study to evaluate ICV administration in the brain. So we gave 4.5 e to the 9th vector genomes into the lateral ventricle of the mouse, shown in that little cartoon up on the upper right. And on the bottom here, I've reproduced the expression level of S1S3 after intravenous administration in the adult mice so that you can see the difference, for example, here in the brain. Pay close attention here to the y-axis where we're seeing basically almost zero expression in the brain compared to on the right after ICV or ventricular brain-directed administration in the mice at birth, where we're seeing up to like 1 copy of S1S3 per cell. We then looked at dosing the intravenous S1S3 at birth. We first started with a comparable dose to what we had given the adult mice, but the neonatal mice did not tolerate that. We then moved to a 10-fold lower dose at birth, and the neonatal mice tolerated that dose. So then because we were looking at a 10-fold lower dose and we thought that might affect the eventual outcome, we wanted to test 3 different groups. One is delivering the intracerebroventricular with the IV therapy right at birth. The other is giving intracerebroventricular right at birth and then giving the IV at 4 weeks, and compare that with our data from giving the IV only at 4 weeks. But one big question about whether you can give gene therapy at birth and then in one route of place and give it in a different route of administration 4 weeks later was, will that result in an immune response against the AAV capsid that would prohibit you from redosing those mice? So we brought the anti-AAV9 antibody assay into our lab, and we assayed the mice in their serum following intracerebroventricular administration at birth to see whether they would develop antibodies at the time that we would dose the intravenous gene therapy. So whoops, I went the wrong. Can you go back? Oh, I can go back. There we go. So this graph shows the antibody titers against the AAV capsid with the vehicle treated, where they have 0. The ICV, so basically treated at birth, so with ventricular, so into the brain gene therapy, plus intravenous gene therapy at 4 weeks. And so at 8 weeks of age, you can see they did develop anti-capsid antibodies after the intravenous gene therapy. But after ICV gene therapy, at 4 weeks, giving it at birth, at 4 weeks of age, at 8 weeks of age, at 16 weeks of age, those ICV-treated mice did not develop any detectable antibodies against the capsid. So we felt that we could dose, it's kind of almost a redose of gene therapy in these mice. This is a graph, like I showed before, looking at the manna-6-phosphate content of the 3 lysosomal enzymes that we've assayed, alpha-mannasidase, beta-hexosaminidase, and beta-mannasidase, in either the mice that were treated with ICV plus IV at birth. Remember, ICV is into the brain, into the brain ventricle, IV is intravenous, or given the ICV at birth and then the IV later at 4 weeks. And the mice that were treated with ICV at birth and then at 4 weeks, later treated with IV, had much better correction of their manna-6-phosphate content in these lysosomal enzymes, whereas we really did not see good correction in the mice that were treated with the ICV plus the IV only at birth and not later at 4 weeks. So we have therefore embarked on a long-term study of these mice. They're treated ICV at birth and then IV at 4 weeks at the dose that we previously gave, the times 10 to the 11th gene therapy. We've also crossed this line onto a mouse line that has it's called LAMP1 with a triple flag tag. Basically, it means that you can immunoprecipitate lysosomes and be able to determine whether we've restored lysosomal enzyme to the lysosomes and whether those enzymes might be manna-6 phosphorylated. Could be something we could potentially explore. And so we're currently collecting mice for proteomics and lysosomal enzyme assays from that line. And we have an ongoing long-term experiment with the mice that are treated, and we have been looking at behavior, bone mineral density, survival. These mice range in age right now from 6 weeks to 53 weeks, so I don't have data to show you as this experiment is still ongoing. So that is all. I'd like to thank all of our collaborators and our support from the National MPS Society, as well as from the National Institutes of Health and elsewhere. Thank you.
Thank you, Dr. Dickson. Questions for Dr. Dickson?
Can you explain the rationale why it works that way?
Why what works what way?
What you just described, how it's better to do it one way versus the other.
Well, what I think, so in terms of the ICV plus IV giving at birth, we kind of thought that might work really well, but the problem is when you go tenfold lower dose and then the mouse grows, that gene therapy given doesn't replicate in the mouse. So that's all you get, and it's now distributed over a bigger mouse. It really is. It might be the dose and the size of the animal.
Other questions?
Hi, Dr. Dickson. So translating mouse data to humans, how would you propose a clinical rationale or clinical protocol in a human setting?
Yeah, so we've started to kind of look at what those endpoints would be. And I think that if I remember correctly, it was a couple of years ago, maybe 2 or so years ago, we put together a bespoke gene therapy application and I think it was submitted by the MPS Society as well as some of the other researchers that we were all involved in. So there are a number of endpoints that could be looked at. I think the clinical program kind of depends a little bit on what outcome measures we see best improved in the rest of the animal studies. We do have plans to move into a larger animal model, and I think that will be informative for some of the choices we make there.
Yeah, I agree. And especially we know AV9 works quite differently in a newborn mice than it does in other animals.
Yeah.
It travels to many other tissues in newborns that you can't get that same kind of efficacy in other larger animals. So I'm also wondering about the capsid and if you've thought about other potential capsids that cross the blood-brain barrier more efficiently, so you could just deliver one route of administration versus two.
Yeah, it's something that we would be very interested in partnering with with someone who has a good capsid. What's nice about the AAV9 capsid is that there's history of it getting into patients, into clinical trials at least. FDA isn't looking unfavorably on AAV9 as a whole at the present. So while it seems to be working, we think that it's a good way to go. But I agree, it would be nice to be able to explore some other capsid ideas.
Thank you.